a result, you get completely normal genotypes. Now, genetic exchange in trypanosoma teeth is actually known for quite a while from Trypanosoma brucei. It has been shown more lately in recently in Cruci, in Leishmania major. It has been shown in this example, but not in this example. So if you look at these taxa here, it seems to be fairly widespread in the clade, but it occurs at some tips of the branch. It does not occur everywhere. And if you also look closely in all these cases, actually, these three cases, they are vector-transmitted diseases, whereas in our case, is a directly transmitted disease. So the genetic exchange does have nothing to do with the fact that you need a vector. It also occurs apparently in normal co-infections in the host. Now, if that happens, and the parasite is spread in a colony, of course, what you're interested in is what happens with the strains that are present. And here is an experiment that has been done by Jutta Ulrich lately, where she actually infected colonies with a defined cocktail of strains and watched how these strains sort of went through the colony in an epidemic rush, so to speak. And what's remarkable is that at the end of the colony stage, the representation is not very different from the beginning, but it starts to differ quite a bit once they jump to the daughter queens. And I recall you that the daughter queens are the transport vehicles that allows the parasite to get through the hibernation into the next year. So that seems to be the stage where actually the available infections are filtered out and actually reduced to some sets with some successful strains, like here strain number, I think it's D, E, and B here, which are quite successful compared to the beginning. And these are the infections that get on into the next season. So it also turns out that the likelihood that these queens are infected and then lose a lot of fitness next year is the diversity of circulating strains in the family they're coming from. So the more strains that there are circulating in the colony here, the higher the likelihood or the chances actually that the offspring queens get infected. So as you can see here, diversity, which is generated by exchange at the end, then is filtered out by selection, and this selection must come about somewhere from the defense system of the host, because otherwise it's the same environment. And this defense capacity, so to speak, determines how likely it is that you infect your daughter queens. So in other words, if you're able to fish out most of these strains, your daughter queens will be less likely infected and be much more successful than if you're not able to fish out the strains. So again, reducing diversity here seems to be a central problem that these colonies have to achieve, and presumably a major player in this job is the immune system. In fact, it turns out that these strains compete with each other. So if you keep them in the medium, then you always have more or less one winner and a loser strain in, let's say, parallel stage competitions. And this is shown here with the solid dots, with the black ones here. This is competition staged among strains, and it's here represented, for other reasons, but it's here represented in terms of genetic distance between the strains, so how related in inverted commas they are, and the likelihood that they exclude themselves. As you can see here in the black dots, there's almost a step function. So if you're below a certain genetic distance, you're actually coexisting most of the time. Above, you start to exclude each other. And now the important thing is, if you repeat the whole story in the living host, so in vivo, the situation changes completely. And what you get in the host is coexistence, whereas otherwise you would not. So again, we have a situation where the diversity of infections is somehow entangled with what the host does, and presumably what the host defense system is achieving. Now, this is the part now of the second part of the talk here. So we're coming back to the question, we have a huge diversity of infections, which seem to be very characteristic in the system. We know how it's generated. And of course, the question is now, what are the hosts doing about these infections? And I remind you that this is an infection that stays in the gut. So the question, of course, is what is the defense system, or what can insects do to defend themselves against parasites in the gut? Now, the answer to that is we don't know very well yet. I mean, there's still a lot of research going on, and it's not very much known. But there's a list of candidate things, like, for example, the production of these reactive oxygen species that may play a role in the gut. There is a possibility that AMPs, so antimicrobial peptides, are locally expressed in the gut and play a role. It is possible that gut microbiota, so the bacteria that live in the gut, play a role in the immune system. And we shouldn't forget that defense is not only a matter, of course, of the immune systems, but it's also a matter, for example, of gut barriers and compartmentalization. So, if you look at that, let me make one brief remark about gut microbiota, because we're currently working at that in much more detail, and this is work by Hauke Koch. What he did find here is a survey of the gut microbiota, so bacteria in particular, in different bee species, bumblebee species, bumble species, in different locations. And this is done by looking at the 16 sRNA gene sequence, which is the standard tool. And what turns out to be the case is that gut microbiota is amazingly of low diversity. We would have expected to find hundreds of species of bacteria. And if you remember, that's certainly the case, let's say in mammals, that you found hundreds, maybe even thousands of species of bacteria that are part of your microbe community. Here in bees, and actually this is the second evidence together with work done by Nancy Moran and her group, and she found very similar things across many different species, bee species like apis, for example, honeybee, and some other species. This is restricted to bumbles, and the message is the same. If you look at what kind of bacteria exist in gut, the diversity is actually quite low, and it is restricted to probably about 10 species, maybe 20, but it's not much more, which is amazing, which is really amazing. Here you see some of these gray areas. Doesn't matter in particular what they are here, but you see here they're actually distributed in this clade, which is firmicutes of bacteria. So there are different groups of bacteria which take part in this community, and that also turns out to be the case, and I show this here, is that if you look at the distribution of this microbiota among different bee species, bumblebee species here, so here each color code is a different bumble species here, which is common in our areas. It is plotted, so the profile, the 16 RNA profile of the bacteria that they take out is taken apart and plotted here in this graph to cluster them. And of course, at first sight it looks like a real mess, but probably you can see here Soroensis, which is one species, is characterized by this cloud here. For example, Pascorum, which is another common species, characterized by this light blue clade. And in fact, if you do the statistic, it turns out that these bee species are significantly different, not all of them in pairwise comparison, but it seems that this low diversity community is also to some degree specific to the host species. So these host species that exist in the same community differentiate among each other by having slightly different bacterial communities inside them. So the difference between species, by the way, is stronger than the difference between localities, like Swiss and Northern Germany. I'm telling you that, I'm not showing data because we haven't yet done the final analysis, because what we now find is that this microbiota plays a role in defense against this trypanosome. It seems to protect them against trypanosome to some degree. And this, of course, opens a whole new can of worms. I mean, we're now going from the host defense system to something that the bacteria apparently add to the defense. And we don't know exactly what it is, but it is not unlikely that this bacteria also produce some antimicrobial peptides to kill off this parasite. So when we're talking about the host defense, of course, we should not forget there could be also commensals, or whatever you like to call them, symbionts, that play a role in this game. And in this particular case, it goes in this direction. Now, coming to the immune defense proper, this is a representation of several different pathways, which I'm sure most of you are familiar with: Toll, IMD, JNK, JAK-STAT pathways, which are some of the classical pathways of insect immune defense. And in gray are genes that we know exist in our particular species here. So the message of this graph is, yes, indeed, we seem to have a full complement of these pathways, and it is likely that the host is using them for defense against this paradigm. Now, unfortunately, the defense of insects against protozoan is something which is also not very well understood, regardless of whether it occurs in gut or somewhere else. So we have been dabbling along our way a little bit and asking ourselves what is likely to happen. And of course, the one method of choice in this particular example, if we know next to nothing, is to try to do some expression studies with candidate genes. So this is a very simple one. We now have a much better library. For example, here, these are infection experiments. So white is always, for example, the unaffected state, black is the infected state across different colonies. Because again, a hallmark of the system is that you have a lot of variation, not only on the parasite side, but also on the host side, and you have interaction, as you will see in a second. And what turns out to be the case is that you get upregulation of radish and hymenoptocene, which is one of the classical standard AMPs of these groups of animals. And if you go back to the graph here, you can see that this speaks, for example, for the involvement here of the IMD pathway that is activated when it is infected by this particular parasite. In fact, it turns out, and this was the first study which we're now following up in more detail, is that if you start to infect these animals with different strains of this parasite, and remember, we can clone these strains, genotypically different on these skewed markers, and if you infect them in controlled ways, in this particular case, you infect a number of different colonies which represent different genetic backgrounds, and then you measure the expression of antibacterial peptides, it turns out to be an interaction effect. And this is shown, I'm afraid, not so clearly visible, but it's the only way you can really see this data. This is the expression of 3 different AMPs, antimicrobial peptides, which are also classical peptides in the group of animals, so defensin, hymenoptocene, abacine. And the colored dots represent the expression level, of course, above standard and controlled and everything, so on, challenged by different strains. So these are strains 1 to 4, different genotypical strains. And if you do the statistic on the whole cloud, what turns out to be the case is that it depends not only on who you are as the host, what background you have. It not only depends on the parasite that is infecting, but it depends specifically, and that's the interesting bit, on the interaction. And that's what we are after. We are after the interaction term here. So it depends on the combination who infects whom. Depending on how this happens, actually, you find an upregulation of these AMPs, in particular hymenoptocene and defensins. These two are connected to IMD pathway. Defensin is partly connected to IMD pathway. It's probably also triggered by something else. So what the message here is, indeed, as you get these infections, you get the interaction not only macroscopically in terms of the end result, let's say infection events per se, but you also get an interaction in terms of upregulation of a cocktail, so to speak, of antimicrobial peptides. And that's what we think now I'm working on, is that the essential ingredient here seems to be a differential upregulation of peptides in a cocktail that then together actually works to contain or eliminate this infection. Now, if you talk about this upregulation and this defense, of course, a classic question, and Marty was using that in introduction, is the cost of defense. I'm not going now down a list of, let's say, repeating all the things that you can see and kind of show there's a cost, but I'd like to illustrate the cost aspect in a slightly different way. It has to do with the diversity of infections that we were talking about. For example, some time ago we figured out that there is a cost, or a trade-off is probably the more correct term in this context, between the diversity infections that you can control and the actual standard measure of general immune responses. So the general measure here is, this is just not working, doesn't matter. Good German to speak here. This is the strength of the general response. So this